determined from long-term catches MRIP data. So
hello, everyone. Thank you so much for coming out to listen to my talk. And today I'll be sharing the work that I've done for my first chapter of my dissertation. I'm a PhD student at North Carolina State University in the US. So the long-term data interest came due to recent warming in the Atlantic along the US East Coast and an interest in wanting to study the phenology and range shifts of some popular recreational uh, fishes in this region. However, these fish are larger and faster swimming fishes, as a lot of popular recreational fishes are, so they're not often caught in bottom trawl data sets, which is what is commonly used for long-term data with fishery independent surveys and often used in climate change studies with fisheries in this region. The recreational species we were interested in were dolphin fish and cobia, and these represented species with narrow thermal preferences, which basically means they prefer the water temperature to be within a few degrees of each other, versus bluefish and striped bass having a wide thermal preference so they can withstand a wider temperature range. And our hypotheses with this work were that there would be phenology and range shift signals in the MRIP data, which is recreational catch data. The catches would shift to earlier arrival dates, and there would be evidence for stock shifting northward. However, these changes would be more pronounced in our species with the narrow thermal preferences. So this data set is from NOAA, which is the federal fisheries agency in the US, and it's called the NOAA Fisheries Marine Recreational Information Program. It started in 1981 and is creel surveys. So we've got people interviewing anglers on docks as they return from fishing and asking questions about their catch and also some user data. At the time of the data download for my project, I have 42 years of data, but this is an ongoing survey. So it's got over 45 years of data now. They use this data to estimate total catch and effort for the fisheries in the US. However, I use the raw creel survey data before those estimations occur. We also had to account for some survey spatiotemporal coverage. Basically, people are not fishing in January in Maine, so they don't survey every state for every month. So we have Massachusetts to Florida from March to December. To model the catches of these species, we had a response variable of species counts, and we aggregated the catch data over year, ordinal day, and latitude. This was because there are so many zeros in this data set. There's millions of observations. So to help with model fit and all those things, we summarized the catches. We also included man hours as an offset term to account for effort, since this is a fisheries dependent data set. And our ordinal day predictor was transformed into a harmonic term, which just means that instead of the model recognizing day as a linear effect of 1 to 365, we transformed it to be a cyclic effect so that December and January are back to back, so you can model seasonality. We did this by taking the sine and cosine of the day or multiplying day by 2 and taking the sine and cosine of those variables so that you could have up to 2 peaks of seasonality, so spring and fall. We used generalized linear modeling due to our response being a species count and compared the models with AIC and checked for model robustness with cross-validation. From this data, we learned that there were phenology and range of signals present within the MRIP data set, which was exciting. However, this is fisheries-dependent data, so we always want to check on changes in angler effort and how that could be impacting the results. So to get at this from the beginning, we looked at the mean man hours fished on the y-axis across space and time. So we've got latitude on the x and time as a decade, different colored lines. Here you can see that around latitude 35, which is like North Carolina, Virginia, there is a lot more mean man hours of fishing happening. However, over time, it has remained relatively stable across the region. When looking at range shifts, we found that species with narrow thermal preferences are shifting northward and species with wider thermal preferences shifted southward or were neutral. Here we've got cobia, one of the narrow species, and bluefish, a wide. We have decade on the x-axis and the weighted latitude on the y. The shaded region is a 95% confidence interval, and then the black line was the observed from the raw data, and the red, the predicted line from our models. You can see that cobia are starting to shift northward over time, and bluefish slightly southward. We were also interested in range shifts at a finer scale, so this was more like the central average distribution here. We wanted to see how it was looking at like the northern and southern edges of the range. So we looked at the percent change of catch from previous decades to the next. That's what's on the y-axis here, decade on the x. And our states are in rainbow geographical order. So red being northern Massachusetts to our pinks and purples being the southern states. The main takeaway here is that while the cobia population has been increasing over time, we see that our southern states have remained relatively about the same, whereas our northern states are having this disproportionate increase in catches, indicating that a northward range shift of cobia may be occurring. In contrast, our species with a wide thermal preference, striped bass, their populations have been on the decline, but we see that it has declined and stayed about the same across our states, indicating there is no range shift occurring. For phenology, we found that species with narrow thermal preferences had more instances of a significant shift to earlier arrival dates at northern latitudes in the spring relative to our wide thermal preference species. Here we have dolphin fish on the left and bluefish on the right. And what you're looking at here on the y-axis is something called a 50% day metric. In phenology studies, it's often when 50% of the fish have passed a certain point, like a river mouth if it was salmon, to find the peak migration date. However, with this data set and these species, we found that they're migrating north and staying north, so they're available to anglers to be caught. So that 50% day was getting drugged later and later in the year, even though the raw data showed that it shouldn't have been that. So to remedy that, we split the year into a first and second time period and calculated that 50% day metric in both of those. So that's what you're seeing here. And we'll focus on the first, so the red. Here you can see all the dots are that day for every latitude, and then we ran a simple linear regression to test for statistical significance if that day had really changed over time. And in these three states, as examples here, you can see this line having a negative slope indicating earlier arrival dates for catches of dolphin fish, and then a pretty flat line and not statistically significant change for our wide thermal preference bluefish. These shifts in phenology led to changes in the number of days spent at given latitudes, and this occurred across all of our species in no apparent pattern amongst thermal preferences. So there were changes in the number of days spent, but the time period that was impacting that number of days was different. Here we've got our species narrow and wide, and then the abbreviated states in geographical order from north to south. The blue boxes indicate that it was an earlier arrival, so that first time period was significant, changing and giving them an increase in the number of days there, whereas the yellow was that they were staying longer, so a later departure, or a combination of the two. We found positive numbers, so longer time spent at all of these latitudes, except for striped bass having two states with a decrease in the amount of time spent there. Again, this is fisheries dependent data, so we wanted to be transparent and look into angler behavior here as well. Here you're looking at decade on the y-axis and ordinal day on the x, and we have a black dot for every day that a catch of this species occurred, with a red circle indicating that they were targeted. We had fishing effort across these time periods, but in the MRIP data set, anglers can report what they were targeting. They often don't report what it was. They can leave it blank. But in the days that we did have it, we have it plotted here to show when anglers were targeting a species versus when they were catching them. And these were all states that had significant phenology changes for dolphin fish, but in North Carolina, you can see that dolphin fish were available throughout these decades, and anglers were catching them or targeting them around the same time period. But in New Jersey, when anglers were targeting them has shifted. So these are kind of confounding variables. So it's hard to disentangle if it's a phenology change from the fish or the angler behavior. But also anglers are smart and they talk or post on social media. And, you know, if the fish are there, they're going to be out there fishing for them. So in conclusion, there were phenology and range shift signals present in this fisheries-dependent data set, and we found that species with narrow thermal preferences were caught earlier and farther north in more recent decades compared to species with our wide thermal preferences. These narrow thermal preference species are responding to climate change in a more pronounced way than those wide thermal preference species. So that might be something to consider for management and vulnerability assessments, and maybe prioritizing species that prefer those narrow thermal preferences. And this study highlights that long-term data is needed to study the impacts of climate change, especially phenology, as this could be a 2 to 3 week shift, and you need long-term data to see those maybe subtle trends. Recreational fishery-dependent data could be a key data source, especially for our larger and faster swimming species like these cobia here. And overall, by better understanding past and current fish distributions, we can help coastal communities prepare for changing fisheries and a changing climate. And thank you so much for listening, and I'll take questions.
Thanks very much for your talk, and we have time, I believe, for one question now, because we need to keep track of the time for the other sessions. Anybody? I have it.
Yeah.
So we recently published a paper, a student of mine did, on redistribution of species due to climate change on a smaller scale than yours, but commercial catches. And what we used for quantify this shift was mean temperature of the catch. I don't know, mean temperature of the catch.
Oh, okay.
So it's a concept of physiology and optimal thermal condition for the species that you can easily calculate. And do you think this can be applied to your data set?
Did that include where the fish were caught exactly? Yeah. So this data set, they don't have the exact latitude and longitude of where the species were caught. It's more where the survey site was. So we had the centroid latitude of the county. So it may differ where that, well, that was why we didn't include temperature in our models, because we don't know how far offshore these catches were occurring. But that would
this
definitely
can change a lot, yeah.
Yeah.
To the oceanographic system that
yeah,
fishing.
yeah.
I see.
One limitation of this data set.
Yeah, I see.
Thank you.
Yeah, thanks.
Thank you.